I'm Helén Tuvesson, CEO of Active Biotech, and I'm happy to present to you one of our core projects, Tasquinimod, which we develop as a new product class for use in multiple myeloma. This is our forward-looking statements. Tasquinimod is an oral small molecule immunomodulator which we develop in multiple myeloma, and there is currently a phase 1b/2a study ongoing in relapsed refractory multiple myeloma. Tasquinimod has already demonstrated clinical proof of concept by significant progression-free survival benefit in prostate cancer, and the safety is well known with more than 1,500 subjects exposed to Tasquinimod. We have a regulatory package of the required preclinical safety, and we have a clinical database from previous studies in solid tumors. There is a full commercial scale CMC documentation available, and we have new fresh IP protection to at least 2035, an orphan drug designation granted in the US. Multiple myeloma develops in the bone marrow, where the plasma cells start to grow and prevent formation of other important cells like the blood cells. This leads to bone pain, fractures, anemia, and other severe complications like renal failure and infections. Survival has increased lately due to several new treatment options available, and the median survival is now estimated to 8 to 10 years from diagnosis. However, patients relapse and eventually die due to resistance to current treatments. Multiple myeloma patients undergo several lines of treatment, mostly combinations with 2 to 4 drugs, but patients relapse as resistance to treatment develops. After 3 to 4 lines of treatment, there are very few options left, and comorbidity and poor tolerability to rather toxic drugs further limits treatment options. There is an unmet need for novel and safe drugs to be included in non-cross-resistant combination therapy. Tasquinimod is a new drug class with a mode of action that is novel and complementary to current multiple myeloma therapies. Multiple myeloma develops in the bone marrow, and the microenvironment in the bone marrow is important for establishment of the multiple myeloma and is also a key driver of relapses and development of resistance to treatment. The myeloid cells in the tumor microenvironment promotes local immune suppression, angiogenesis, and tumor growth. Tasquinimod inhibits the function of these myeloid cells by inhibiting S100A9. And thereby re-establish the immune function and decrease the growth of new blood vessels, leading to tumor killing. Tasquinimod's mode of action is novel and is different to that of the 4 main classes of standard therapy used today in multiple myeloma. There is an urgent need of efficacious and safe combination regimens, including drugs with novel mode of action, to mitigate drug resistance. Tasquinimod, with its novel mode of action, good tolerability, can fit very well into future combination strategies. The myeloma market is a major market. It's expected to continue to grow, and there is a substantial market potential for all lines of treatment. New treatments and treatment strategies have substantially improved survival lately, but still patients are dying from the disease. To support deeper and durable responses and overcome treatment resistance, patients are, as a standard, treated with combinations of drug products from available classes. As illustrated on this slide, with a prevalence of 1 million patients treated per year, about 280,000 are estimated to have failed first-line treatment, and a further 350,000 are estimated to have failed both first-line and second-line treatment, and today are in the third-line plus segment. Our data indicate that Tasquinimod is well tolerated and can be combined with the available drug classes. Tasquinimod therefore has the potential to expand over time from an initial position as the third-line plus, which is similar to the current study population, to earlier stages of treatment. Estimating the commercial potential of Tasquinimod at this early stage is of course difficult, but taking a position given that it's a new product class, we estimate a commercial potential of more than $1 billion. Tasquinimod as monotherapy has anti-myeloma activity in preclinical models. The upper panel shows that Tasquinimod prolongs the survival of mice that have been challenged with mouse myeloma cells. The cells are injected into an immunocompetent mouse, and they then grow in the bone marrow of the mouse, just as myeloma cells do in humans. As can be seen, Tasquinimod significantly improves overall survival in these myeloma-bearing mice. The lower panel shows another example in which human myeloma cells are implanted under the skin of an immunocompromised mouse that accept human cells. Treatment starts once the tumor has been established, in this case 10 days after cell injection. Treatment continues throughout the whole study, and as shown in the lower panel, the growth of the tumor is significantly reduced in mice treated with Tasquinimod. Tasquinimod also demonstrates evidence of synergistic activity with other myeloma agents. In this study, mice have been challenged with human myeloma cells. Treatment starts 18 days after tumor injection, and the mice receive either Tasquinimod alone, the proteasome inhibitor bortezomib alone, or a combination of these two treatments. As shown here, the combination of Tasquinimod with bortezomib reduces myeloma cell growth more than Tasquinimod or bortezomib alone. This synergy supports the fact that Tasquinimod's mode of action is complementary to proteasome inhibitors. Active Biotech has a collaboration with the Wistar Institute in Philadelphia, and a team led by Dr. Yulia Nefetova has performed these experiments. Based on our compelling preclinical data and the previous clinical experiments with Tasquinimod, a clinical study was initiated, and the first patient was dosed in August this year. The study recruits relapsed refractory patients after at least one prior anti-myeloma therapy and is conducted in two parts. The first part assessing monotherapy effect of Tasquinimod, and the second part a combination of Tasquinimod and oral standard anti-myeloma regimen. Primary endpoint in both Part A and Part B is safety and tolerability, and key secondary endpoint is preliminary efficacy by overall response rate. The study is carried out in an academic partnership with Abramson Cancer Institute in Philadelphia, with the principal investigator being Dr. Dan Vogel. The phase 1b/2a study is ongoing according to plan, and currently we expect the first safety readout in second half of next year. Following established safety, an MTD dose expansion cohort will be started, as well as the Part B combination part of the study. The final readout of monotherapy Tasquinimod is expected second half of 2022. Our key priority right now is to advance the clinical study. In parallel, we will prepare for the next step clinical development. Important correlative analysis of the study biosamples will be performed at Wistar. This analysis aimed at support further understanding of Tasquinimod biological effect in the disease. It has been a pleasure to update you on the progress and plans for Tasquinimod, and I look very much forward to update you as the project advances. Thank you so much.